Homework should be available to you guys. If it is not, it'll be up this weekend. I don't remember what time I was going to have it come up, so just be patient on it. Okay. Forgot. I got to pull up PowerPoint. This one. That's not the right one. I clicked on the wrong thing, didn't I? Yep, that's not it, guys. Hold on a second. It's because it keeps moving me to the wrong one. All right, this has to be it. My complaint to eLearn is they need to make the boxes longer so that you can actually read them, because I really struggle with that. They also used to have it color-coded and like icon-coded, and they don't do that anymore either. PowerPoint was orange. It was easy to see. All right, stop being whiny. I'm blessed. All right, so can anybody tell me? All right, so we stopped with the stress response. Our major stress hormone, we're going to talk about it in a minute, is cortisol, right? Where's cortisol released? Yes, specifically. Very good. So that's going to be the adrenal cortex. I have to use this for my notes today. And more specifically, it's going to be the zona fasciculata. Very good. We're doing a little bit of a review because I can. All right. What is going to be released from the zona reticularis? Yeah.
Sex hormones.
That's going to be your sex hormones. That's going to be primarily your androgens. You probably know what I'm going to ask next. What's relieved from the zona glomerulosa? Yeah. Very good. That's going to be aldosterone. And as a class, aldosterone controls what? Sodium levels and potassium levels. So it's going to retain sodium, which causes a retainment of water, and it's going to excrete potassium. All right, good job, Hannah. Good job, Hayden. Very good. All right, I'll ask some more questions once we get into the stress response. We're going to finish that today, and then we're going to move into blood. So along with blood, we're going to talk about its components. We're going to talk about the life cycle of an RBC, and we're probably not going to get into white blood cell basics. I didn't quite get there with the last class, and we don't need to. We just need to get through like 22 slides. I have some extra ones on here today. That was just in case we had time. Reminder, your exam, I was supposed to say Monday. Don't look at that. It's supposed to be Monday, 1/27, so don't get confused. It's 27th. And then Sawyer, you want to talk to them for a sec?
Oh, yeah, sure. Morning, guys. Our first study session was on Tuesday. I thought it went really, really well. Thanks to everybody that came out. I ended up sending all the stuff in the group later so you guys can study on your own. We're going to have another one this coming Tuesday. It's going to be the same place, same time. So you're all invited to that. And then we'll probably have one the day before or 2 days before your test, which is on the 27th. So,
yep.
Good.
So probably a couple more study sessions. Please take advantage of all of them. Endocrine system isn't very easy, but we are taking that exam with the blood system. And the blood system is, I don't know, it's a little bit easier in my opinion. You probably know a little bit more about it than you did the endocrine, if I had to guess. So we started talking just a little tiny bit about stress. Stress is really important. It is vital to your survival. We talked about how, you know, if you're not stressed at least a little bit, you're probably not going to be motivated to do very much. We went through this already. So let's talk about our different types of stress. So we can have psychological stress, and we can also have physical stress. So a psychological stress is going to deal more with your mental. Do you feel in danger? Is it causing you to sweat? Are you starting to panic? Are you starting to shake? It could be due to a personal loss. That could be of a human being. Or it could be a possession, something was destroyed, you had a house fire, something of that nature. Also, it deals with anger, fear, as well as guilt. And most of those emotions are going to be controlled around the amygdala. That's like your fear center of the brain. Another type of stress is going to be physical. And this is probably more or less, I don't know. This is what I encounter more on a daily basis. So this is going to be your temperature extremes, becoming way too cold or way too hot. I mean, if you get way too hot, you're going to start going into heat stroke and whatnot. You guys all did a case study on that last semester if you had any. It also can result from infection. That can be some type of bacterial infection, viral infection, also injury. And injury can be, you fell down, you hurt something, you broke something, but also, if you have a break and your body can't repair it, what do you typically go into? To try to fix it. Go to the doc to have surgery, right? So surgery is kind of like an injury that's going to be created to try to fix the problem. Anybody that has been through surgery knows that's going to cause quite a bit of stress on the body, doesn't it? Exactly. So that can cause a release of cortisol and all kinds of different factors. Also, lack of oxygen. This is not on my bucket list. It may be on some of your all's, but I do not plan on trying to hike Mount Everest before I kick the bucket. All right? Not something I want to do, but some people, that's like their lifelong goal. What happens when you try to climb to this high elevations? Yep, your oxygen levels are going to start to depreciate. Some people literally have died trying to get to the top of Mount Everest, and that's just not in my plans. But anyway, so that's called hypoxia. And so your body is going to respond to that to try to, with all these things, we're going to respond to the stress to try to bring our body back into homeostasis, right? Now, just kind of thinking about it, we're not going to talk to our neighbor, we're going to think. So which type of stress do you think is more, we'll do this, I'll give you a 5 for Friday, if you get it right and you explain to me why. Which type of stress is more detrimental to the body over the long term? Yes, Hannah.
Psychological, because it can interfere with different hormones and stuff, and it messes with your day-to-day actions because it can cause your body to go into physical stress.
Great, you got your point. All right, so it is psychological stress, and it's for pretty much the exact same reasons that Hannah talked about. It really messes up with every single organ system, especially your endocrine system, right? All hormones and everything that is there. So let's talk a little bit about your response to stress. Oh, sorry, I didn't put the objectives, the last 2 objectives back up again, but they were being able to describe the differences and kind of compare psychological and physical stress, and then also being able to describe the general adaptation syndrome, which is what we're going to talk about right now. There are 2 main stages to it. The hypothalamus, pretty much your control center up in the brain. He's going to be controlling your response. And so 2 main stages. One is going to be the alarm stage. And this is going to happen pretty much on at least a weekly basis with everybody. It's your fight or flight response. You wake up late. You're running late somewhere. Somebody scares you. Somebody tells you terrible news that you're actually not going to care about the next month. That's going to be your alarm stage. So it's immediate, not going to last very long. But you have those sympathetic impulses coming through the body. So last semester you talked about sympathetic and you talked about parasympathetic. Sympathetic, some things it's going to do, it's going to increase blood glucose as well as fatty acid concentration because you're going to break down some lipids. Why are we increasing blood glucose?
So that your body can produce ATP.
Yeah, for one, so your body can produce ATP, and also your brain pretty much runs on one type of sugar, and that's going to be glucose, right? So we got to get that to your brain because you got to think about what you're going to do next. Also, heart rate, breathing rate, those are all going to increase. That's essentially to get blood and nutrients to your body. Blood pressure is going to go up with that. We're going to dilate air passages. Why are we doing that?
Breathe.
So what are we doing in breathing? We're inhaling what? Oxygen and exhaling carbon dioxide. We're getting rid of all those wastes so that we can continue to make ATP and do whatever we need to do. We're also going to shunt blood to the skeletal muscles. That's like what we talked about last time, as far as being important to get oxygen nutrients there so they can keep moving. And also we're going to increase epinephrine secretion. Where is epinephrine released? Very good, the adrenal medulla, along with norepinephrine. So in this response, epinephrine is going to intensify and prolong each one of these responses. Now, hopefully, you're going to be able to get yourself calmed down and get through the situation, and then the alarm stage is going to be over. However, if it perpetuates, you will move into the next stage, and that is going to be the resistance stage. It is slower and longer lasting. Now this is where we start getting into the production of cortisol, which we've talked about some of its effects. So this is a CRH, ACTH, cortisol pathway. Who can tell me what these abbreviations mean? Okay. You almost got it. I'm going to have to say it now because you were so close. So this is corticotropin releasing hormone. This is adrenocorticotropic hormone. You're very close. Watch the endings of those. It's not a huge deal. But just keep that in mind. I actually, if I'm being honest with you, I looked it up again this morning to make sure I didn't get them mixed up. But anyway, so this one is coming from the hypothalamus. This one is coming from the anterior pituitary, because any hormone that is released from the hypothalamus is only going to act on the anterior side of the pituitary gland, right? It's the nervous stimulation that'll affect the posterior. And what two hormones are coming from the posterior? Very good. Antidiuretic hormone, or ADH, as well as oxytocin. But on this pathway, so hypothalamus, anterior pituitary, and then cortisol is being released from the zona fasciculata because it is a glucocorticoid. And so essentially, cortisol secretion is going to increase, so cortisol is going to increase in the bloodstream. From there, this is going to be important because, well, we're going to steal glucose, we're going to steal glycogen from other areas of the body, and we are going to give that to primarily the brain so that we can think and try to get through this situation. So cortisol, glucagon, and growth hormone are going to mobilize energy sources for other tissues and organs. That's also going to go along with this. It's essentially just trying to bring you back into homeostasis. Something else that's going to occur along with these high glucose levels is going to be ADH, which we just said covers the posterior pituitary, and we're going to have renin coming from the kidney. Antidiuretic hormone is going to cause you to retain water, and then renin is going to cause, with the RAS system, release of aldosterone, which retains sodium, which is going to do what? Retain water. So it's a bifold focus. Also, with those increased sugar levels and also increased salt levels, what do you think that person's going to want to do? Drink. That's another response that's going to come to this. So with all that water being retained or coming in, that person's going to start to swell up. And also they're probably going to start getting into edema. This is not a happy stage to stay in. Hopefully the body will be able to overcome and get out of that stage. Otherwise, that person is going to enter into the exhaustion stage. And that's really not going to be a fun stage. Ash, I got to write down your name before I forget. All right. So with the exhaustion stage, this is going to begin after, it's individualistic, but after months, at least 1 month of being in the resistance stage. And so this is going to start to cause a wasting due to depletion of nutrients in the body. Yeah, a really good example of that. If we're stealing glycogen and glucose and stuff from your muscles, what's going to happen to your muscles? They're going to atrophy. They're going to waste away. Then you can get into electrolyte imbalances, right? There's a certain condition, so you've got anorexia and you've also got bulimia. And this can play into a stress response. It's not the sole thing. But people that are bulimic, what do you think's happening is they're throwing up all the time. What are they losing? They're losing electrolytes. They're also losing water. A ton of other stuff too. Anyway, so they're losing all that. A lot of times bulimia is coupled, they're trying to lose weight. It's coupled with exercise. So it can be coupled with like exercise-induced anorexia. There are a number, you can look at the stats yourself, I don't know the stats, but there are a number of bulimics that, they start getting into this electrolyte balance, and then they're still going out on all these real long runs, and they're sweating out even more. commonly what will happen, the heart, electrolyte balance is really important for the heart. Sodium and potassium, we'll talk about it more when we get into the cardiovascular system. They end up having a heart attack when they go out and do that. So electrolyte balance isn't something to mess around with, along with water. All right. Now, we also then have immune system suppression, which is not good. If your immune system is suppressed, can you fight disease very well? No, not very well at all. And if you think about it, if you, usually during around exam season, it's easier to pick up colds and viruses and things like that. Why is that? They're studying for all these finals. They're kind of getting stressed about it. Now, typically people usually come anyway because they don't want to have to make it up later, or they can't make it up later. But this is due to immune system. It can be due to immune system suppression. And immune system suppression doesn't have to wait until the exhaustion stage, but this is highly correlated with this stage. And so the effects of all these are basically due to long-term oversecretion of cortisol. And if it is not taken care of, it can result in death. That image did not come from your book, but I think it does a really good job of talking about the different stages and kind of what's happening in that, or displaying it. It doesn't really talk about it. Oh. All right, this is another chart looking at the general adaptation syndrome. What I need you to know for the exam regarding the general adaptation system is looking at those 3 stages that we just talked about and being able to understand, all right, what is going on in the alarm stage, what's going on in the resistance stage, and what is going on in the exhaustion stage, especially knowing which hormones and if the fight or flight response is involved, things of that nature. You should know that. You might also want to know that you're going to do the 5 for Friday thing. All right. So these are major events. You definitely do not have to memorize this. The only reason I put this up here is because I wanted you to realize with the general stress syndrome, look at how many endocrine organs, glands, and their hormones are involved in this syndrome. And also just keep in mind, when we talk about the general stress syndrome, do you guys know what a syndrome is? Talked about it before. Think about metabolic syndrome. It's not a disease. It's a category. There's usually, with a syndrome, there's a variety of different symptoms that can be involved, and if somebody is categorized as having X amount of them, they're diagnosed with having that syndrome. So with this syndrome, it would be they're experiencing X amount of symptoms that we were talking about on the last few slides. So just FYI on that. All right, guys. Great job. Done with chapter 13. So homework is due on Sunday. Yay. A lot of you guys have that done. I'll plan on converting those over again after Sunday from Connect to the Great Book. All right. Do you guys have any questions on the endocrine system? If you're kind of struggling with some of the glands, what they're producing, downstream effects, I highly recommend that you go to Sawyer's study sessions because he is very good and he can help you out with that. You are also welcome to come to my office and I will help you the best that I can. From there, like I said, we're going to be moving on to what's, in my opinion, a little bit easier chapter, easier things to digest, things you're probably a little bit more familiar with, and that's going to be looking at the blood, which will lead us into the cardiovascular system after your first exam. That's chapter 15. In lab this week, it'll couple with what we're talking about in here because we're going to look at blood typing. We're going to look at what happens when blood types are improperly transfused into carriers. Unfortunately, we are in West Virginia. When I was in Florida, we actually did blood typing of like my students' blood, which is really cool. I'm not allowed to do that here. So I have to use synthetic blood. But that's okay because we're going to be kind of investigating. You guys are going to do some different crossings and figure out what unknown A and unknown B are. So it should be kind of fun. Anyway, that's coming up. We'll also learn more about the RH factor. So, but for today, we have some objectives we want to be able to meet. Number 1, describe the general characteristics of blood, looking at the components, function, as well as its importance. Then we want to distinguish between the formed elements and the liquid portion of blood. The liquid portion of blood starts with a P. Any idea what it is? Oh, that was good. Plasma. All right. Next, describe the origin of the blood cells. and their lineage. We'll talk about that in a minute. We'll talk about my quotation marks. All right. Next, explain the significance of RBC, red blood cell counts, as well as their use in disease diagnosis. I mean, if you think about it, it seems like every little whip stitch, if you go to the doctor, if there's something wrong, they're always wanting to do a CBC. What's a CBC? Very good. That's a complete blood count. Does it almost? Yes, I'm not even going to say it does. It just is going to contain a red blood cell count, and from there they may get the hematocrit and all that from it. All right, next we will get into this a little bit, the life cycle of an RBC and the control of their production. Last class stopped at what happens at the destruction of a red blood cell, so we'll try to make it there today. If we don't, it's okay. We'll deal with it. All right, so let's look at the characteristics of blood. So it's the only type of connective tissue that has a liquid matrix. If you remember from last semester, we talked about 4 major types of tissues, right? All right, so we had connective tissue, epithelial tissue, muscle tissue, and nervous tissue. Very good. But blood is going to be your liquid connective tissue. All right, really important. Used for a variety of different things. For instance, transport of nutrients, including glucose, oxygen, carbon dioxide, waste, all of that. And it's going to regulate processes to maintain internal stability. It's also going to maintain the stability of interstitial fluid, and it's going to help to distribute heat. And it's done by water. If you guys remember back, I know you got to remember a lot from AMP1, but thinking about water, one of its properties, and specifically dealing with the blood, is going to be a high specific heat or high heat capacity. Exactly. And so, because of water's properties, it's going to take a lot of energy to change the temperature to either increase or decrease. If water is a major component of blood, that's going to help to also stabilize blood temperature. And you have blood running, hopefully, from your head to your toes, right? So that's going to help for you to be able to maintain body heat. Thinking back to water, another important characteristic of that was the heat of vaporization, which also helps with temperature regulation of the body, but that's less to do with the actual blood. That's more with water. All right, so blood's really important. We define that. Blood volume is going to vary with your body size. Most of you guys are going to have a lot more blood than I am, just because of size differences. It'll also vary based on changes in fluid concentration, so whether you are dehydrated or whether you're fully hydrated or you're working on it, like Chase. All right, it's also going to change and fluctuate based on changes in electrolyte concentration. If you eat a really salty meal, what's going to happen? You're going to be thirsty, which is going to cause what? Water retention. So it can work in a twofold way and with what you're talking about. Exactly. Also the amount of adipose tissue. Males typically have more water than females, which is going to increase blood volume. That's due to muscle. But we're not getting into that. All right. So blood is going to be about 8% of the body weight. The adult blood volume is going to be between 4 and 5 liters in a female and 5 to 6 liters in a male. So about a liter or so difference. You guys know how much about a liter is. Half of a 2-liter Coke bottle. All right. So let's look at the components. So blood cells it's a misnomer. So we're going to call them formed elements. All right. Most of these guys are formed in the red bone marrow. And we have 3 major types. We call them formed elements because in reality, out of white blood cells, red blood cells, and platelets, only the white blood cells are actually cells. So we are going to come to find that our red blood cells, or the erythrocytes, actually lose their nucleus at about mid-differentiation stage. They kick him out, and that's so that they can carry more oxygen. Fun fact, they also don't have mitochondria, so the only way that they can produce ATP is going to be through glycolysis within the cytoplasm. Platelets are also not cells. They come from, we'll talk about it later, but a megakaryocyte, and it's huge. And so as he tries to squeeze through the different blood vessels, little pieces break off. So blood platelets are literally just cell fragments, and they deal with blood clotting. So another term we use for those is thrombocytes. Again, misnomer, cyte is at the end of that meaning a cell. Thrombo refers to clotting. They're not a cell, but it is a formed element. So the correct term to call these items are going to be formed elements. And they're going to constitute approximately 45% of the blood. The other 55% is going to be blood plasma. I actually really like this chart because I think it lays everything out very nicely. We're going to talk a little bit more about plasma components later. We're going to talk about red blood cells, white blood cells, and then platelets first. So this is going to be a series of lectures. But we can see some of our different plasma components: electrolytes, water, proteins, waste, nutrients, and gas, and breakdowns from that. Over here, major formed elements, and we also, we see the white blood cell percentage. If you look at it as a whole in blood, out of the formed elements, 95% are RBCs. Much smaller percent are your fighters, immune system components, about 0.1%. Platelets is about 5%. Even though that 5% is fairly small, platelets are really important. If you don't have enough platelets, what are you going to struggle doing? Yep, you're going to struggle recovering from any sort of cut because you're not going to be able to clot very well at all. All right, so most of you, I'm sure all of you, have had blood drawn at some point in your life, probably some more than less. It's going to be really important. Usually they do that if they think that something is wrong with you. That sounded wrong. That's what I mean. But anyway, also just sometimes it's going to be on a routine type of basis, just to make sure everything is in the right proportions. That'll keep you healthy. So whenever they take blood, a few things has to be done. So whenever you go to that site, they're probably not going to be actually testing your blood there. It has to be sent somewhere else. So they're going to have to keep the blood cold. Also, once they take your blood, we're going to need to centrifuge it to break it down into its correct component parts. Because a lot of times they're just, they'll look at the hematocrit and the red blood cell content portion of it, but they'll be looking at the plasma for any abnormalities of proteins and whatnot there. So we're going to have to spin it down. But before we spin it down, we have to put in something like heparin. Do you know what heparin does? That is one way to describe it. You could say that it thins the blood. Good, but do you know what another term for that? Huh? It can be used for that. They put that, sometimes people have to use that. That's what I was looking for. Very good. It's an anticoagulant. So without that, if they drew your blood and put it into a little tube, it's going to coagulate. And then you can't run blood tests on it. You can't really do anything with it at all. So some of you guys know, I did research on mice, and we did mouse surgery. Surgery was fatal. Anyway, we took their blood, and so for like a regular mouse, we called them wild types. They were C57 mice. They were black. So I would do cardiac puncture while they were under anesthesia, and I would get about 1 ml of blood from them. Before I did that, I would take the 1 ml tube, and I'd put, I can't remember how much it was now. I think it was like 10 microliters of heparin. Put that in the bottom. That way, whenever I drew their blood, I would immediately put that into the tube, shake it up, it wouldn't coagulate. At that point, we had like a little mini centrifuge. The blood banks have, huge ones. Ours is a little mini one. And so it spins down at a certain RPM, depending on how much blood volume is going to be there. And at that point, you're going to get layers. Those layers are going to be based on the density of the components that are in there, along a little bit with size, but that plays a role in density. All right, so when you spin blood down, you're going to get 3 major layers. The largest layer is going to be, plasma. And that's going to be, like we talked about before, approximately 50%, 55% of your blood. Plasma is going to be this like clearish, kind of a little bit tinted yellow, straw-colored fluid matrix, and it's going to be found in the top. Percentage, not in order. The next percentage down is going to be 35 to 46% in females, 40 to 54% in males. It fluctuates just a little bit. And that's going to be your red blood cells. If it falls too much below that, you're anemic. And that's not good. You need to get help. but that's going to be found at the very bottom of the centrifuge. And then if we look and we take the percentage of actual RBCs, that's what we're going to call the hematocrit. Last but not least, we have the layer where we're going to find the white blood cells, or the WBCs, as well as our fragments, so the platelets. And another term we can use for that is going to be the buffy coat, and that's going to be found in the middle, and you can tell it's itty bitty baby tiny, less than 1% of the blood. Really cool. A lot of times, like if you're doing experiments, if you want to extract DNA, you can do that from a variety of different ways. When I did it with mice, you can do this when they're little. It's humane. You clip the end of their tails, and then you put that in kind of proteases and other components, and it breaks it down, and you can get to the DNA. But you can also do it from blood. And where you would extract the DNA, it has to go through a lot of purification processes, but you can get it from the buffy coat. So just cherry on top of the information you're getting today. Oh, and that's just a blood smear, and we'll be looking at some of those in lab next week under the microscope, and I'll also put, I think, a virtual lab up where you can do a blood differential and you can like count them out and stuff. It should be kind of cool. All right, so how do we get these formed elements or shells? That's going to be through a process called hematopoiesis, aka the origin of formed elements. So that's how we are going to eventually get to erythrocytes, which are RBCs, leukocytes, which are WBCs, and thrombocytes, aka platelets. All right, so the blood cells. I was doing this all last period too. All right, so blood cells are going to originate in the red bone marrow, and this is going to be from their grandfather cell, which is going to be, you can call it one of two things. It's the hematopoietic stem cell. Hematopoietic coming from hematopoiesis, stem referring to its undifferentiated. It has not come to its mature state yet. Or it's called a hemocytoblast. So hemato and hemo both refer to blood. Very good. Cyto refers to site, and then I'll tell you, blast refers to immature. So our grandfather is the hemocytoblast. I know the chart looks kind of complicated. It's really not. We're going to break it down, and we're going to talk about every single one of these components before we get done with this chapter. But let's start out with the hemopoietic stem cell. So they are going to give rise to more specialized cells depending on the growth factors that are present. So that's going to designate what we're going to get to. The next two cell lines that are going to come down from the hemocytoblast or hemopoietic stem cell is going to be the lymphoid stem cells, and those are going to give rise to lymphocytes. So you've probably heard of things like T cells and B cells and memory cells and whatnot. Those are all coming down through this line. So that is through your wonderful lymphoblast. Again, lymphoblast is an immature cell till it gets to B or T cells. We'll hit those pretty hard when we get to the immune system. We'll talk about there are different types of each one of those, even at that. You can see this at the bottom. All right. Pretty much everything else is coming from the myeloid side. And so those are going to give rise to, eventually, the erythrocytes, our platelets, as well as our the white blood cells that are not lymphocytes. They're going to be on that side. All right. But we're going to talk about the RBCs first, aka the erythrocyte. So we've already defined cyte means cell. What does erythro mean? What color are they? That was good. They were red. I got at least half the class to say something. All right. They're in a biconcave disc shape. This is really important for what we are going to be transporting, as well as so they do not stick to the sides of your vessels or to one another, because we don't really want that. We don't want any clots in our vessels. That could lead to something like a stroke or a heart attack. All right. So RBCs are going to consist of approximately 1/3 hemoglobin, and that's going to be important for transporting oxygen. hemoglobin is also going to be a means of helping us transport carbon dioxide as well, but we'll get to that later. All right, so we have a couple of different states, I guess you could call it. We have oxyhemoglobin, and then we also have deoxyhemoglobin. So, yeah, and I'll leave it at that for a second. All right, so these guys, as a mature cell, they are going to lack nuclei, because we'll go into the stages in a minute, but before they become what's called a reticulocyte, they kick out the nuclei. They do not have mitochondria either, but they can produce ATP through glycolysis. They also cannot divide. Therefore, this process of hematopoiesis, and we'll go specifically into erythropoiesis, which is the further differentiation into an erythrocyte. That's going to be very important, because how else are you going to give rise to new ones? And class, where does this occur? Where does hematopoiesis occur? Very good. Red bone marrow. Class, where does it occur? Oh, there's more of you. All right. All right, so I'll take a hand on this one. Would you expect there to be a difference in color between oxyhemoglobin and deoxyhemoglobin, and if so, what is it? Mikey, you're the first one. Yes, very good. So oxyhemoglobin is going to be your deeper, kind of a brighter red, whereas your deoxyhemoglobin is going to be more towards a purplish color. Anyway, yeah, very good. And that's for obvious reasons, because of oxygen. You think about it, in your veins, blood is gonna be a much different color than when it is exposed to oxygen. It's gonna turn bright red. Very good. Wrong way. Okay. So, looking a little bit closer at our red blood cell counts. So this is looking at specifically the number of RBCs, and we have to define the units. Otherwise, it would be arbitrary, the actual number that you count. So the units that we use is either cubic millimeters or microliters of blood. And as you can see up here, males are going to typically possess more red blood cells than females or children, but children kind of go right in between. And for their size, they produce a ton of RBCs. This is also why when babies are born and then through the first little bit of their life, so they have more RBCs. That means they're also going to break down faster. They're going to have more breakdown of them. When red blood cells break down, which we'll look at, I guess, next week on Wednesday, they're going to end up, as they break down, they're going to release biliverdin, which is going to be converted into bilirubin. What color is bilirubin? Anybody know? It's not red. It's yellow. Therefore, babies are more susceptible to jaundice. Very good, you guys got it. So the yellowing of the eyes, fingertips, skin in general. Now, that can be taken away. They can be put under UV light. That helps to break down bilirubin. And it's not a big deal because it's temporary. However, if an adult has jaundice, that's a whole other story, because that's probably due to something like liver failure, and that's not good. All right, I digress. All right, so red blood counts are really useful in diagnosis of disease, as well as evaluation of their progress. Let's say that somebody is anemic, so we might do just kind of a baseline. You know, what is your RBC level? Or we may have to look at hemoglobin or something like that. Depends on what type of anemia. All right, so then a month later, after we're using some sort of intervention, maybe it's an iron pill, maybe it's an iron infusion, we'll look about a month later, whatever the doctor wants to do, and we're going to check it again. So it's helpful in looking at the progression of a disease, hopefully in a positive direction. And just overall, it's important because it's reflective of the oxygen carrying capacity of that individual. So specifically with red blood cells, this is going to be through a process called erythropoiesis. I got your name down, Mike. That's what I was doing. All right. So this is, again, going to occur in the red bone marrow. And so the process involved. So you're going to have, likely, if it's going to occur, a low oxygen content. So lower oxygen saturation. And that's going to stimulate We usually talk primarily about the kidneys. This is what we mentioned in the endocrine system, but the liver can also do it too. And it's going to release erythropoietin. Remember, talked about it with Lance Armstrong. All right, so within a few days of the secretion of EPO, it's going to be your abbreviation for erythropoietin, many new red blood cells are going to start to appear in the blood. Now, as you probably figured, because of how many negative feedback mechanisms there are in the body, this is a negative feedback mechanism. So that's what we can see over here. Low blood oxygen content. Kidney says, I got to start putting out erythropoietin. Liver might help out with that too. Goes into the bloodstream. So erythropoietin's target cell is going to be at our skeletal system. It's going to be within our bones. So that's going to stimulate red bone marrow to be producing more red blood cells. Once our oxygen levels rise, because we have more red blood cells, the system is going to shut down. Kidney and liver are going to say, all right, time to take a break again. All right, so a red blood cell is born. Ding, 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 yay. And it's going to begin to develop. So we go from a hemocytoblast to an erythroblast. Notice still immature blast. Now at this point, this is where he loses the nucleus. It's going to disintegrate. Then we get to the reticulocyte, and last but not least, we have our erythrocyte, or red blood cell. The average lifespan is about 120 days. You got to think about it. He's moving all around. He's squeezing through different passages. It's not going to be that long. Cells have a variety of different lifespans. If you think about it, when we talked about skin cells, their lifespan is not very long. We got to produce skin cells pretty frequently. And within your mouth, that's really, think about all that, the endothelial cells. Whereas something like an egg cell within a female, where she's born with that, that's going to last about 50 years. So really big range within the body, depending on what type of cell it is. Muscle cells, I think it's like 15 years, something like that. And neurons, it's pretty much going to be a lifetime, right? Because if you destroy your brain cells, you're not coming back from that very well. So be careful what you do to your body. All right. So there are specific nutrient requirements for erythropoiesis. We need vitamin B and folic acid. These are going to be helpful in DNA synthesis, along with growth and division of all of the cells. Obviously going to be important here. We also need iron. Iron is going to be required for hemoglobin synthesis. It's also absolutely essential for you to even be able to carry oxygen. You have your heme groups, and there's going to be iron associated with each one of those in a hemoglobin molecule. Okay. So we just talked about the function of each one of these substances that are going to be required in order to go through the process of erythropoiesis. And as you can see, all of the sources of each one of these is going to be dietary, because you're going to take them in, and as long as everything's working properly, the small intestine is going to be your major site of absorption for anything that you're ingesting, your nutrients. So they're going to be absorbed by the small intestine and then go to their respective areas. Now, iron is really special. You don't have to intake as much as you would if the body did not recycle it. So when red blood cells die, your hemoglobin is going to break down into heme, and it's going to break down into globin. From there, we're going to take our iron, and we're going to send that back to, it can be a few different places. One place can be the red bone marrow, and that's to help us. So that's, it's helpful, so we don't have to worry about taking in as much iron. Some people have more trouble with that than others. I personally have struggled with eating red meat. The only red meat I can really eat without getting sick is venison, deer. I grew up on that. So I'm iron deficient if I don't take iron supplements. And I don't eat enough liver. Liver is a really good source of iron. I'm just going to leave it at that. It's not my favorite. All right. So anemia is a condition in which the oxygen carrying capacity of the blood is reduced. And this is going to be due to a deficiency of one of two things: red blood cells or hemoglobin. I have a chart that we can look at. Here, don't pay attention to the arrow. But we can see some red blood cells that are, they're fairly healthy. It's kind of cool. Do you know why they're white in the middle? No.
Concentrated because of the biconcave.
That's it. It's the way that the light is reflecting off of it because they're a biconcave disc, so that's why it looks white in the middle. Now, does this look like a healthy RBC? No, looks like it's sickled. So if there are a bunch of those, then that person likely has anemia. It could be due to sickle cell disease or one of these other conditions up here. So we can see, I gave you two different categories. It can be due to anemia, can be to a decreased red blood cell number, or it can be due to something with hemoglobin. And that could be either decreased hemoglobin concentration or abnormal hemoglobin. Now, abnormal hemoglobin is usually due to something genetic, such as sickle cell anemia, which I have another slide on that, or something like thalassemia. Just in general, decreased red blood cell concentration could be due to bacterial, viral infections. If somebody has an improper blood transfusion, there's going to be coagulation there. There's going to be a lot of RBCs that are destroyed. Also, deficiency of one of the factors involved in creating red blood cells. We'll get into more of the intrinsic factor when we do digestive system. Also, certain medications. If somebody has cancer and they have to go through chemotherapy or radiation for another purpose, that can also lead to anemia. And I have the different types over on the side too. The most common one that I hear about is iron deficiency anemia. I have that, and I know several of my students have it too. All right, this gives you information on sickle cell disease. This is really dangerous. Again, it's from a mutation that codes for hemoglobin. So without a lot of hemoglobin, can that person move oxygen around very well and get it to the tissues? No, absolutely not. And so that can cause pain, for one thing, over time if we're not getting enough oxygen there. Pain in that instance, like, it's a good thing and a bad thing. It tells your body, hey, there's something wrong, but it's not fun. And unfortunately, with sickle cell disease, it's a perpetuating downhill cycle. Because with that, we have a sickling, all right? So that can cause the cells to conglomerate around your blood passages, and that causes a narrowing. If there's a narrowing, then what's going to happen to oxygen levels? They're going to drop more, which is going to cause more sickling, which means they're going to stick more to each other. And it's a downhill from there, unless we can get that person treated. If it's a child, we can give them antibiotics to help prevent infection. We can also give hydroxyurea, and this actually causes the body to produce fetal hemoglobin. That's helpful because fetal hemoglobin is actually better at binding to oxygen than adult, so that can help with this disorder. Transplant of bone marrow might be helpful, but that can be dangerous, along with umbilical cord stem cells. And then we have other up-and-coming ways. It's controversial because use of stem cells and also changing somebody's genome, it kind of gets into ethics there. All right, this is talking about the death and destruction of red blood cells. We will stop here. I put some questions at the end of this PowerPoint that we are not obviously going to do today, but you may use these as a study tool. All right, if you can't get the answers, let me know. All right, reminders. MLK Day is Monday. We do not have class, so I'll plan to see you on Tuesday if you have me for lab. Kate Duffy, your TA, is presenting next week. You can support her, and that's all your assignments due coming up. You guys ready to have a great rest of your Friday?
Yes.
Let's go. All right. If you have questions, stay by. Let me know.